Hi, Gianna, it's Miss Agar. I'm going to give you some direction on this lactase case study so that hopefully you find it easier to navigate and complete. I have confidence you can do this even without being in the class, so let me just give you some tips. Okay, so first of all, this article was to help build background knowledge. So the Neolithic Revolution essentially is where societies transitioned from hunter-gatherer lifestyles to domestication of plants and animals during the agricultural revolution. So the bottom paragraph is what we, in terms of biology, would focus on. So if you read over this, what it is pointing out is that after the westward spread of agriculture into Europe, there was dairy farming that Europeans tended to practice, which clearly left a stamp in DNA. So prior to the arrival of domestic cattle in Europe, prehistoric populations weren't able to stomach raw cow milk. But at some point during the spread of farming into southeastern Europe, a mutation occurred for lactose tolerance that increased in frequency through natural selection, thanks to the nourishing benefits of milk. So judging from the prevalence of milk drinking gene in Europeans today, as high as 90% in populations of northern countries such as Sweden, the vast majority are descended from cow herders. So what we're going to focus on in this case study is this mutation that occurred that enabled particularly Europeans who were dairy farmers to digest milk into adulthood. The videos that are attached here will give you background on the milk that mammals produce and the mutation that also enabled us to digest it as adults. So key point, mammals typically don't drink milk as adults, but this mutation that occurred within the human gene enabled people to start to digest lactose as adults. So normally mammals wean their babies off of milk, but essentially people can now continue to drink it even past childhood. Okay, so two things you're going to need for this case study is the case study like packet and then this data sheet, genotype and phenotype chart. So we'll start looking at the case study. So I would highly suggest you read everything, but I want to give you an overview of the sections and some clarifying detail. So if we focus on this diagram first, lactose is a sugar in milk that is broken down by the lactase enzyme into glucose and galactose. We can compare people that produce the enzyme as adults to people that don't. And so what you can see on this diagram, if you have the enzyme lactase to break down lactose, it becomes glucose in the bloodstream. If you don't have the enzyme, the lactose is not broken down. It continues to be fermented by bacteria in the gut, which will produce gases and acids that make it essentially uncomfortable for people that are lactose intolerant. Okay, so this term lactase persistence means that the enzyme persists into adulthood, meaning adults would continue to produce the enzyme because their gene enables that, and then they can digest the lactose. So if you're tolerant, you're persistent. If you're intolerant, you do not persist to make the enzyme. All right. This research was done on individuals by having them drink milk and then test their blood glucose levels to see what happens. So if you digest milk, if you can break down the lactose, it should become glucose in the bloodstream. So notice on this graph, all of these people where their levels of glucose are rising should be digesting the lactose, which is becoming glucose, and therefore glucose levels are rising. If you can't digest the lactose, your glucose levels essentially don't rise. Here's a quick video clip just to show you how that test would work.
You're allowed to breathe in between, it's all right.
If my body is still making lactase, my blood glucose will shoot up. After I drank the milk, here's what happened. No doubt about it, my lactase enzyme is still working.
Okay, so these few questions are about those people on this graph, so you can work through that. This is a section that you wouldn't necessarily be able to do without having the lab materials. So this is just a pre and post test using milk. We want to see if there's glucose in the milk to begin with and compare it to what if we add the enzyme, what happens to the amount of glucose in the milk. So to give you a little bit of an idea of how this test works, you would take milk. It doesn't really matter what kind of milk. If you have skim milk, 1%, 2%, none of that would matter because that's the amount of fat in the milk. We're talking about the amount of sugar in the milk. So essentially, if you take the amount of milk that you have and you use these glucose test strips, you're going to be able to measure the amount of glucose in there. So we do a pre-enzyme and a post-enzyme test. Now, where's this enzyme coming from? It's this Lactase pill. So if you go to the pharmacy, you can actually buy these Lactase pills, which have the enzyme. So if someone's not tolerant, they can take these pills, which will help them then digest the milk. So in terms of how to measure the amount of glucose, these test strips are color-coded. So this would be the color for milk on its own without the enzyme added. And then this is a change in color on the strip that you then have to compare the color chart to in order to determine the amount of glucose. So notice the color change. So if you have the test strip color chart here, you can tell that initially there's no glucose in milk, but if you add the enzyme, the concentration of glucose rises. So when we see this increased concentration of glucose, it's because that enzyme that was added from the pill, so you can crush up the pill and dissolve it in the milk, and then the enzyme will break down the lactose in the milk into glucose. So in the first place, there's no glucose in milk, and afterwards, there is glucose in milk. On your lab, the results that we had would be 0 concentration of glucose in regular milk, and then the concentration went up to like 1,000 milligrams per deciliter. So you could jot down 1,000 as the increased concentration. So again, the results would be due to the enzyme in the Lactase pill. So they also sell at the grocery store what's called Lactase milk that has already had the enzyme added to it. So if you see Lactase milk or you see Lactase pills, they have the enzyme that will break the lactose down into glucose. Okay, now in terms of genetics, what is actually happening? In a minority of adults, a mutation in the lactase switch prevents the lactase gene from turning off. So if you look at this diagram, a normal mammal as a baby, the switch to produce the lactase enzyme is on. When mammals wean their children, typically the switch turns off. So if the switch is off, then the gene is not read, the enzyme will not be produced. So if you think about that relationship between DNA, RNA, and a protein, the enzyme is a protein. The gene codes for production of that enzyme protein. What actually reads the gene is RNA, and then that will be utilized to produce the protein, just like a chef. So if DNA is the cookbook, the gene is one recipe, RNA is the chef that reads the recipe and assembles the amino acids to produce this enzyme, which is classified as a protein. So the mutation that occurred that enables adults to digest the lactose is actually in the switch. It keeps the switch on so that adults can continue to produce the lactase enzyme. So then this question is about the genetic cause of lactase persistence, which you should be able to explain using the diagram and the information here. All right, in terms of a pedigree chart, we're going to look at comparing the pedigree from a Finnish family in Europe to, on the next page, an Asian family. So in terms of the pedigree, if you're not familiar with them, they're based on like generations through a family. So at the top, these would be the older generations, and near the bottom would be younger generations. So for example, if you were in generation 5, your parents would be generation 4, your grandparents would be generation 3, Your great-grandparents would be generation 2, and going back even further in time to the first generation. There's the key here, so you can see that females are circles, squares are males. If it's shaded, they're intolerant. If it's white, they're tolerant. So you're just making a claim here. So think about the pattern that you see in the pedigree chart regarding tolerance or intolerant patterns through inheritance in the family lineage. In terms of why it would be useful for individuals within the same family to be studied, if you think about your family members have mostly similar DNA. Of course, there's differences between individuals, but it's much easier to compare the genes of people that are mostly the same in order to find where the differences are. So if you have a family member that's tolerant and a family member that's not tolerant, you might be able to hone in on where that mutation is, causing one person to digest milk and the other to not. All right, then we're going to take a look at that Asian pedigree. So essentially, there are many more intolerant people in this family. So again, you're just using that pedigree to make some interpretations and a prediction for the future of the Asian gene pool. Okay, there's a lot of information on this page, but when you get to question 11, this is where you're going to need to look at the data from the genetic data sets, and the next page is the phenotype data. So the phenotype data is based on people who drink milk. Do their blood glucose levels rise? Genotype data is based on, do we see the mutation in the genes of the people that were studied? Be very mindful, cautious, aware that these studies, so for example, questions 11 is going to ask you about Greece. So the Greek population here only had 100 people sampled, and the study was done in 2009. But if you look at the study that was done on the Greek population for phenotype data, they used 600 people, and that study was done in 1974. So the people that they tested by having them drink milk are not necessarily the same people in the test where they looked at their actual genetic mutations. So just know that some of the studies for phenotype data are not necessarily the same people in the genotype data, even though they're part of the same population. It doesn't necessarily mean they sampled the same individuals in both studies from each place. All right, on this chart, these frequencies are in decimals, but know that they would be percentages. So if you're looking at 0.3, that would actually be 30%. So you can just mentally convert them into percentages like 84%, 13%, 0%, 25%, 45%. So ignore the decimal essentially. So questions 11 through 17, you're just using that data to make some interpretations. When you get to question 18, this is where you're going to need the Google Sheet. So again, on Classroom, there's another post. Google's being real slow right now. There's another post on Google Classroom for the data sheet. So if you open up this lactase case study data for the map and you open the Google Sheet, it's your copy. So you can just simply input the data that is on that paper copy. So same information, you have the continent, country, population, number of people sampled, number of people with the gene, without the gene. So what you needed to do essentially was just fill in this column using this sheet for the genetic data. So only putting in the percentage. So if you're going to put in the frequency of the Somalian population on this sheet, you can just put in the number 30. Don't worry about the decimal or the percent symbol, just put in the percentage. You could even skip this column and just go to the country data because it's the same thing. So you either are going to input it on this sheet and copy paste, or go to the country data sheet and just put the numbers in there. Right. This is going to give you the ability to make a map. So you highlight all of these columns. Once you have those numbers input and you go to insert chart, you can get a map. So you're going to change the chart type to the GeoMap chart. When you have all of the numbers in there, it's going to color code reds and greens initially to give you the percentages. So the brighter the green, the higher percent frequency, meaning those are the places where people are more tolerant and persist in their lactase production. The brighter the red, the lower the frequency, and then you'll see some in between color. It might be a little bit gray or in between green. There's no data for North and South America, so don't make claims about that because there's no data to make a claim based on. So all of the studies have been done within the Eastern Hemisphere. So just initially ignore now North and South America. All right, once you have this map, you should then be able to answer some additional questions on the case study. So question 18, you're making a claim based on the map. The evidence that supports your claim is going to be based on those, reds and green regions and outliers, things that don't obviously fall in line with your claim, but it's still possibly relevant. All right, question 19 is an interpretation based on migration patterns because there was no data from North America. For question 20, in terms of global hotspots, those are the regions of high frequency. So think about how many areas that are bright green. And this is not a question that there is an answer to necessarily, but how could we determine whether people in those hotspots have the same mutation or if each mutation is unique? So imagine you had 4 hotspots. Are all 4 of those places are people in those 4 places, do they have the same genetic mutation, or would there be 4 unique mutations? So essentially, to answer this question, you'd have to compare the mutations or the genes of the people in each area to determine if their sequences are the same. So if they have the same mutation, the genetic sequence would be the same. If there's unique mutations, their genetic sequences would be different. So you just have to compare the genes of people from each region in order to determine if they have the same mutation or unique mutations. Okay. In this section, visualizing the color pattern is what you want to focus on. So in this diagram, when we think about chromosomes, they come in pairs because we get one from mom and one from dad. So when it comes to the lactase gene, it's on chromosome number 2. We just have 2 copies of chromosome number 2. So there's 23 pairs in the human species. So we have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, 2 number 5s, all the way up to number 23. So we have 46 chromosomes total, but they all come in pairs. So notice this person here is colored red because this red spot is where the lactase persistence mutation is, meaning this individual has the gene for lactose tolerance. After several generations, the number of people with that gene increases, assuming that that helps this individual survive and reproduce, then the gene can get passed on to offspring. So over a handful of generations, more people end up with the gene as it was passed on through reproduction. After many more generations, notice the pattern where the color pattern gets a little bit more mixed up. So here we call these homologous chromosomes because the banding pattern is the same. So this person also has homologous chromosomes, and so does this person. But over many more generations, the banding pattern is not the same on both chromosomes because people continue to reproduce and genes get more and more mixed up over longer periods of time. So the red band is more common after many generations because the individual that had the mutation survived and reproduced and passed it on, and then more people continue to inherit the gene as more offspring are produced. But after many, many more generations, the combination of genes gets more shuffled or mixed up due to reproduction. And so when we see the homologous chromosomes like this, it's after a handful of generations. When we see heterozygous chromosomes like this, it's after even longer periods of time. That can actually help researchers determine when mutations originated in populations of people. So on this next graph, you have the African population compared to the European population. The homozygous region is the similar base sequences. So when we compare Africans to Africans, there's 1.8 million base pairs they have in common, versus when we compare Europeans to Europeans, they have 1.4 million base pairs in common. So Africans have more similar base sequences than Europeans. That brings us back to this. If there's more similarities here, this would be like the African population. And after longer periods of time when genes get more mixed up, this would be more like the European population. So the European population has fewer similarities, so they have more of their genes mixed up. So the European genes that were studied must have been around for longer periods of time than the African population. So the mutation would have occurred in European populations before it occurred in African populations. What that essentially means is that Europeans developed this mutation and ability to digest lactose before the African populations. So when you're looking at the global map and you see a hotspot in Europe, they actually have a unique mutation compared to the African population, where their mutation was totally different and independent, occurred at a different time, and is actually a different mutation. But they both resulted in the same effect of being lactose tolerant. Okay. I highly suggest you watch this film before you move forward, although you could watch it first before you even start this case study. You might want to preview the film and then watch it twice. It could be very informative at the beginning or at this point for the additional evidence that you need to understand the whole scenario. Okay, at this point, you shift into focusing on a different enzyme. So this one is called salivary amylase. Read all of this over, study the diagram, and look at the data. Focus on this graph, which shows you the number of copies of a gene that an individual has. This is to make the salivary amylase enzyme. So the salivary amylase enzyme helps people break down starchy foods rather than lactose sugars. Starchy foods, where do we find them? In things like potatoes, rice, squash, starchy vegetables, wheat products. So if you are a population that tends to plant your foods and you eat more crops than you do drink milk, you might have more copies of this gene. So if you have more copies of the gene, you can make more of the enzymes. So for example, you have one copy of the gene, you make one enzyme at a time. If you have two copies of the gene, you can make two of the enzymes at the same time. If you have three copies, you can make three enzymes at the same time. So how does that really benefit somebody? If you can break down more starches more efficiently, you get more energy for survival. You also have the ability to better digest another food source, so you get more nutrients. And again, if you have more opportunities to eat different types of food, and that gives you more nutrients, you have more energy, you're more likely to survive under different scenarios. So honing in on this graph and then honing in on this graph at the end. So you can see here, populations of people that have high starch diets are also associated with more copies of the gene, so they're making more of the salivary amylase enzyme to break down starchy foods. This is a summary of all the concepts, and you can attempt to answer question 29. It is wordy, but if you focus on what happened across the world over time, how are people related, but also how have we changed as a species? Whatever you put here is good. Anything you learned. And this is very simple. Write a question or make a prediction. There's no right or wrong here. So the last two questions, you're attempting to summarize everything and also make a prediction for the future. Okay, that's a lot of information. I hope it makes as much sense to you as possible. But you could, again, watch that film before you start and then work through the questions, section by section. Feel free to email me if you're still struggling, and I'll attempt to clarify again.